We have guest speakers.
Welcome, Dr. Beattie. We are very, very glad to have you, very excited. We are Ideas. We are a new club with our focus is for ASU online students in the sciences. Our biochemistry program is only about 2 years old. The biological sciences, I think, is only a year older than that. And so we are building the student society aspect, the student support aspect to a new program. It didn't exist until this point. So one of our goals with this group is to provide opportunities to online students that you would get in a more traditional campus, which would be guest lecturers, guest speakers, people coming through academic services, career planning, pre-health, whatever that would mean, and coming and talking to the students and being like, hey, look, this is what you can do with your degree. This is what's available to you. You might not have heard about this. Or just people coming through and talking about their experience going through the degree program, further education, going into private industry, going into academia, whatever that might be. So our goal here is to have people come talk to us virtually through Zoom to get the same kind of experience, because we would benefit from the same guidance. It's just not a structure that's currently set up to integrate us well into ASU's student life aspect of that. So we are doing it ourselves.
Yeah, makes good sense. Nice.
Yes. Your professional and personal background is impressive, and we are very excited to hear about your journey, your choices, and your experiences, because it seems from your CV alone that you've had your foot in everything, in a sense.
Yeah, that's an advantage and disadvantage. So
well, we'd like to hear about that. So
okay. Well, I was born in no. How do you want to do this then?
I think, Sam, probably the best way to go about starting is maybe start with, we have your CV and we have shared that CV with the organization, so many have had an opportunity to look at it. I'd say maybe start with, how did you choose the graduate program direction that you chose, and how did you choose food specifically and regulatory compliance? Maybe start with that, and we can kind of see where it goes.
Sure. So you have to realize that I've been in some aspect of the food industry, if you will, for 45 years. So my father was an academic. He was on the faculty at Iowa State in hotel and restaurant management. We owned a hotel up in the Colorado, San Juan Mountains of Colorado, not very far from Arizona. So, I've been around food all my life. Well, all of us have been around food all our lives, right? But what happened is, and because it's chemistry, right? My first real job, other than detasseling corn, was working in a hospital kitchen. And it's microbiology too. And so when I was 16 or when I was 17, or I think I was probably a junior in high school. They switched, I washed pots and pans, and they switched sanitizers on us. You wash pots and pans in a 3-compartment sink, and the last sink is filled with sanitizer to kill the microorganisms on the utensils and whatever. Well, they switched sanitizers. They went from a chlorine-based sanitizer to an iodophore and an iodine-based sanitizer. And what we learned then, or what I learned, is that I could turn mashed potatoes purple. Okay, and you're all familiar with that starch reaction, right? They titrate out until you hit a blue endpoint. Well, I learned that I could turn mashed potatoes purple with the sanitizer, and that got me kind of, you screw around a lot when you're a kid, and I wound up screwing around a lot. But as it went on, there are other aspects of it that got me kind of interested. And in undergrad, you kind of fuss around, and frankly, I fussed around a lot. I mean, if anyone ever tells you that kids with poor GPAs can't make it in the world with advanced or get advanced degrees, don't believe them. It just takes extra work. I think my first 2 quarters at Iowa State, I had a 1.5 GPA. But then I came across analytical chemistry, and from there on, it was kind of like, wow, this is really interesting stuff. And then I got into the microbiology aspect of it. And what had happened is I was interested in microbiology and I was going to go that way. But I had friends who were in food science who were studying this exact same stuff that I was studying. And the kids in microbiology, the students in microbiology, weren't getting jobs. But the people in food science and technology were getting jobs, and we were all taking about the same courses. So I thought, well, this makes better sense. So I went off and got a degree in food technology. The point then became that, what do you do with that degree? And I went and worked for Kraft for a year, and then worked in management for a year with a different company before I kind of decided that I needed to go off to grad school and get a real job, or it just was time. And so I was interested in microbiology, so I wound up at South Dakota State looking at dairy microbiology. Where you all learn about cheese and yogurt and all that stuff. Well, what I studied was actually a little bit off, because one of the leading causes of food spoilage is mold growth on the product. And so we were looking at why certain cheeses don't mold. And once again, we go back to the chemistry of it and the biochemistry of it. And what we look at is Limburger cheeses and highly aromatic cheeses, they don't mold very readily. And the reason they don't mold very readily is because there's a bacterium that grows on it that produces a very strong demethylase. It effectively takes the sulfhydryl group off the methionine amino acid and releases it as methyl mercaptan, or methyl disulfide. Reacts really quickly with itself. And what that does is that gives that Limburger cheese that rotten, very aromatic smell. And frankly, it's very prevalent also in cheddar cheese. So what we have is we have the microbiology here, as well as the biochemistry associated with it. And then we also have the instrumental analysis that went on with it to look at how to actually measure the evolution of methyl mercaptan from cultures of Brevibacterium linans. And then the challenge studies that went on with that against various molds that were prevalent in cheese manufacture. So it's kind of like pretty neat. But so that was pretty fun, and I enjoyed working in that area. And what happened then is when I got done, I was offered a couple jobs with starter culture companies, microbiology companies that make cultures for cheese manufacturers, yogurt manufacturers, people like that. And frankly, I just that was kind of too much canned research. So the woman who got me involved in dairy microbiology back in my undergrad, a woman named Bonnie Glatz, she offered me an assistantship at Iowa State for my PhD. And I wanted to do that, kind of, but I didn't want to go back to Iowa. I kind of grew up in Iowa. I thought, nah. So what I did is I took an assistantship out of Oregon State instead. Prettier for sure, and a lot more fun. But out there, I was really non-directed. I mean, I screwed around a lot. And what happened is I had an opportunity to go out and work at the Oregon Marine Science Center on oysters. Back in the 1980s, the molecular techniques that we know about now were pretty rare and pretty difficult to do. So what we were looking at is we were looking at a rapid method to determine the health of oysters. And it had to be a pretty fast test, because oysters, first off, they don't grow very fast, but we needed some sort of marker that we could use to determine their health. So what we used is, you guys may be familiar with a enzyme called ornithine decarboxylase, ornithine decarboxylase is one of the key enzymes in the pathway to produce polyamines. And polyamines are compounds that help stabilize DNA in the cell. So what we were going to do is we were looking at going after that target, but we were looking for not the enzyme per se. We were going to go after the message. So we were going to go directly for the messenger RNA, which was really pretty difficult. So, excuse me, this is what I'm going to tell you. What we did is for about 8 months, what I did is I went out to oyster farms and collected oysters, or they provided them, and then I took them back to the Marine Science Center. Now, you know how you tell a male oyster from a female oyster? It's pretty hard. So what we would do is we'd open up oysters, or I'd open up oysters and look at the gametes through a microscope, and look for sperm or egg. And once I found a sperm or male, what I'd do is I put them in a wearing blender and grind them up, and then freeze them overnight. So I'd kill the sperm. And then the next day, what we'd do is I'd take 20-odd oysters and lay them out in a heat table, and not a water bath, if you will. We had like 5 different types of seawater, sterile seawater. We had all those different kinds at the Marine Science Center. And so what we did is, or what I did is I looked at them, and when you start warming up the water around oysters, they start venting. They start opening their shell and pumping more water through it to kind of keep themselves cool. And what you do is you take that vial of the ground up male and squirt it into the oyster. And colloquially, I was working on a PhD on masturbating oysters in many ways, because eventually what happens is the oyster will start releasing gametes. And what you're looking for is, as soon as you see that, you pull the oyster out and allow it to release its gametes. Now, males, males, just whatever it is. But females, when you pull a female out and isolate her, it's fantastic. They'll produce 140 million eggs per one spawning. And so what we then did is we were looking at ways to sequence, get a defined development process. You take the female out and then you fertilize all the eggs at the same time. And what that allows you to do is get a time measurement of as the oyster develops, you can actually measure the ornithine decarboxylase enzyme activity in those oysters, and get a pretty good idea of how the development works. So that was kind of our baseline. And what unfortunately happened is we ran out of money. So it stopped there. So that was difficult. So that's one word of advice as you guys go into grad school, and if you go in further into doctorate and things like that, or wherever, you kind of want to go into a lab that has money, okay? Because otherwise you're kind of, not having money is not very much fun. And so I went back to school, or back to campus, and worked with my eventual there was a group there that was heavily involved in starter culture work. And I wasn't so interested in starter cultures, but I was interested in, at that time we were looking at fish oils. Fish oils were coming up and being very actively pursued. And so okay, so there's that. So went back and started thinking about fish oils because of a lipids class. And we had to write a project for that class. And what that project was based upon my undergrad advisor's research on what are called oleaginous yeast. Oleaginous yeasts are effectively obese yeast. And what they'll do is they'll grow on a carbon source, and they'll in the absence of nitrogen to produce cell mass, what they'll do is they'll accumulate the carbon as oil droplets. So Earl was able to get some of these yeasts to grow on lactose or milk sugar, and he could get up to 60, 70% cell mass as oil. And that was pretty interesting. So the concept we came up with was, and this was a class project, was to look at could we produce omega-3 fatty acids using oleaginous yeast or fungi. And back in 1986, 87, that was a pretty hot topic. What subsequently happened is I took the idea and wrote a grant proposal, which was funded through the U.S., through at that time, the U.S. dairy industry. And they funded me for 3 years for $75,000 to develop a oleaginous fungi that would grow on lactose. So what the 3 years working on that were effectively looking at ways to clone in lactose utilization into the primary organism back then was, or a mold called Saprolegnia parasitica. Maybe some of you guys have seen salmon when they come back from the ocean and they're swimming upstream, and some of them have white spotches all over them. That is Saprolegnia parasitica. It's a fungi that grows on fish, but it produces about 25% of its oil as omega-3, either primarily eicosapentaenoic acid. So that was kind of fun. And I worked, back then, you got anyone who's done electrophoresis maybe in their labs, you have these lovely little boxes that they have. And back then we made our own electrophoresis box out of plexiglass. So it was really a primitive time. Our PCR machine that we had in the lab was a series of pipelines and covered about an 8x10 section of wall instead of the little thermocycles we have now in labs. So so it was the day I got a blue mold colony, because I actually got the lactose gene into the organism, was a pretty happy day. And I drank many beers that night, and it was good. So, are there any questions so far? So, what my life is you look at life, or not look at life, but you look at things and you say, hey, here's an idea, and here's an application for that idea. Let's go pursue it. I love research for the sake of research, but geez, there's so many things out there that we can look at to make things better. And that was one of the things that we thought we maybe have a better market for lactose, which is it's left over from cheese manufacture, and there were warehouses filled with this stuff. So that was what we were doing. We're trying to use lactose from the cheese industry to make omega-3 fatty acids through a biological transformation. And it's all because of my undergrad professor, Earl Hammond. And so it was pretty entertaining. So,
Sam,
yeah.
Oh, sorry. Go ahead. I was gonna say, and then with the time that you spent in research, maybe just kind of change this a little bit. What made you pursue going into working in food in the industry in for example, quality and regulatory compliance?
Well, here's the deal: is that my undergrad, all my degrees so far are in food. Okay, and I used to work in a toxicology group and a biochemistry group, and I was always considered low man on the telephone pole because I was working in food, even though we were doing fairly elegant research. Okay, that was kind of unfortunate. But the point is that The first love is to use food for some practical purpose. And so when I got done with all my degrees and postdoc, what effectively I was qualified to go into biochemistry or anything, but the fact is my true love was food and food science. So, the first faculty position I took was at North Dakota, and even there, it was, there were problems there that needed to be solved. And so, what you look at is you help try to solve the problem. So we, and what we were, a lot of the work we did in North Dakota was on beer manufacture and barley, looking at how mold toxins get formed in barley before or during malting for beer manufacture. So looking for a way to solve that problem was how you stayed in food, okay? And, beer is food, and beer is a science, and wine is an accident. Just remember those things. So, so anyone can make molds go, or grapes go bad and ferment, but to get malt or barley into malt and hops into beer is a little bit more technical. So
Sam, what's distilling in whiskey?
what's distilling in whiskey? Well, distilling in whiskey is actually important because distilling, you do start with some sort of fermentation, right? And so, and you do wind up adding some flavoring, right? So, but still, distilling, that also, you can let grains rot and you get mash for distilling, right?
So you're saying you're a beer drinker?
On those mistakes.
What?
There's $1 billion industry on those mistakes.
There is. Also.
And you're saying you're a beer drinker?
Oh, of course. I used to be a whiskey drinker, but that gets really hard on you, especially when you drink tumblers full of it. So the idea here is then that you go into the food, and I'm in the food industry, okay, and all the research is kind of accessory to that. As you saw from my CV, we've done a whole lot of different kind of research. But my last job was at Iowa State and in academia, and even there, and all through my undergrad, or not through my undergrad, but through my my PhD, I worked in industry, working with organic food processors. Back in those days, it was relatively new. And have you been through an organic audit, Eric?
Yes, I have. And actually, I saw the Oregon Tilth auditor. I've been through many of those.
Right. So I was probably the first organic auditor, food processing auditor in the country. But working with those guys, what you find, and working in the dairy industry, you're the most fun you have is when you're on the floor working with watching what the processes are, and it's fun to watch that because it's just it's a process, and it's you go into Genentech or any of those big pharmaceutical companies based in bioengineering. Most people are not going to go out and work in R&D for those companies. Most people are going to go out and work as a fermentation technologist or a manager of the fermentation technology. So understanding how those processes work is really fundamental. And if you look at it, frankly, if you look at how Genentech and all those guys manufacture a lot of their stuff, it's a lot like making beer or anything like that. It's a simple fermenter design. Pumping is very similar. It's all very similar. And importantly, as is in the food and dairy industry, sanitation and making sure the equipment is absolutely spotless and free from microorganisms is critical. So, what was happening is food safety was evolving, so we got involved in food safety at the academic level. And then my last job at Iowa State was working as the food safety extension person. So what I wound up doing a lot there was actually working directly with food processors on assuring that the food was safe through their processing. And and then the side, my research was working directly with my undergrad advisor, a guy named Earl Hammond, who was 82 years old and still maintained the lab. He's probably one of the premier oil chemists in the world. And we eventually, when my last grad student had developed a feed supplement to encapsulate omega-3 fatty acids, and we'd feed it to dairy animals to prevent biohydrogenation of the polyunsaturated fats within the rumen. So, and what we were seeing is an increase in the omega-3 content of milk. So once again, it's an interesting application to solve a problem. We're making milk healthier, right? And so we had a patent application filed for that. So anyway, but what I found at Iowa State and what I found is in academia, that I was much more happy working in industry than I was working with industry than working as a faculty member. I think that some of you maybe have experienced this, where you run into an arrogant ass of a faculty member who really doesn't have a whole lot of time for you and isn't really interested in he's or she is more geared towards a research orientation rather than towards a student-focused education orientation. And what I was seeing is people who I really respected, who were fantastic teachers, would never get beyond the associate level, associate professor level, because they weren't productive in research. They published periodically, but they just weren't bringing in enough money, or they needed a couple more publications to get that promotion. And they had tenure, but they were still being they were like second-class citizens in many ways. And frankly, I didn't want to play with that game anymore. You go to faculty meetings and you'd sit there and listen to pontifications, and it got to the point where I just said, fuck it, or excuse me, but bottom line is that's what I said, and I left. And one of the consulting groups that I had, one of the groups that I was consulting with, had offered me a position as director of technical services, which included regulatory affairs. And so I jumped at it. And frankly, I've never really looked back. I mean, this was 10, 11 years ago now, or 10 years ago about. And, I frankly never looked back, because first off, the salary is I'm right now, and probably 3 times what I would my colleagues at the university were making. And then college professors don't get paid a whole lot, guys. And then secondly, it's a hell of a lot more fun. It's stressful. It's difficult. But I tell you what, when you're sitting across the table from a regulatory authority or a regulatory person, and the regulations better than they do, and they're trying to rum something up your and you're sitting there telling them, no, here's the citation, and they start scrambling through their code of federal regulations looking for it. It becomes once again an application. Regulatory is nothing more than an application of laws to what we're trying to do. Granted, we're all on the same page, producing a safe food product, but how we get there sometimes is regulatory affairs. Our FDA and USDA folks sometimes don't quite understand their own regulations and how to interpret them. So anyway, that's the bottom line is, what we do in food safety and quality is directly related to chemistry, biochemistry, and microbiology. It's just an application that's a little bit different. So, and then the regulatory affairs people, or how you get around that, or how you work with them to make them understand what you're trying to do. That make sense?
Yeah.
Yeah.
Thank you
So
for sharing your personal journey as far as how you kind of got to the point you're at, which is you are a divisional director of food safety and quality at Hearthside.
Hearthside, yeah.
Hearthside. What
Yeah.
does that involve day to day? I personally have no prior
Oh,
familiarity with the food and regulation industry.
sure. Sure. So the food industry is pretty heavily regulated, right? And so USDA handles the meat and poultry for the most part, and the FDA handles all the other stuff. And so on a day-to-day basis, what I have is I'm on. Our company is very big. We have 38 food processing plants, and what we have is we have plants that manufacture bars. For example, you're all familiar with Nature Valley bars, for example. We make those, okay, for General Mills. We package a lot of foods. For example, I have a box of cheese nips downstairs in my pantry that we package those. Someone else makes them, but we package them. And then we have a group that is highly perishable. So that group is they're the high risk group because the food is refrigerated, it's not stabilized, and it spoils, and it's prone to various pathogenic microorganisms. So it's ready to eat. It gets no heat treatment oftentimes, and you consume it. Next time you walk into a Starbucks and you purchase a bistro box or a panini or something like that, it's likely that one of my plants manufactured it. Okay, and what then you look at is we're absolutely critically concerned about a couple different organisms. Listeria, you're all heard of Listeria, right? I mean, we have massive recalls for Listeria. And so what we do is we look at that, and our requirements are to prevent Listeria from occurring. Now, methods of detection for Listeria in this country are down to 400ths of a microorganism. So what we do is the regulations are very strict on how much Listeria monocytogenes can be in food, and effectively it's zero. So on a day-to-day basis, part of my job is to work with our processing plants on how we avoid having food being contaminated with this environmental pathogen, Listeria monocytogenes. So a couple of weeks ago, and this is really cool stuff, guys. I mean, this is such cool stuff. Um, you know, last month I was in one of our plants and we were having a Listeria problem. And what we were using to help us detect is the microbiological methods for detection of Listeria monocytogenes take 2 to 3 days. We do swabbing for the microorganism. We swab the environment looking for the organism. And we send those swabs off to a testing lab, and they do the microbiology. Well, a company called Neogen has come up, you all know what ribosomes are, right? Ribosomes are the protein factories for the cell. Ribosomes are composed of proteins as well as ribosomal RNA. Ribosomal RNA, if you guys haven't looked up a guy named Carl Woese, look up Carl Woese, W-O-E-S-E. Carl Woese was from the University of Illinois. We have 3 domains of life now, right? We've got eubacteria, we've got eukaryotes, and we've got the archaea. The archaea, the eubacteria, and the eukaryotes all have ribosomal RNA, and they have sections of ribosomal RNA that are identical between the 3 domains, hyper-conserved regions, and they also have hyper-variable regions. So what this company has done is, in order to detect 1 or 2 cells of Listeria, you have to go through a process called enrichment, where you allow the organism to grow up, and then you can detect it. What they've done is each cell of Listeria has tens of thousands of ribosomes and tens of thousands of copies of ribosomal RNA. What they did is they developed a method to go directly at that target, and because there's so many units of it, they can get a signal out of that. And instead of taking 3 days, it takes an hour. So what we do is we swab and we do there's a PCR reaction type thing, and we get a result that gives us the indication of a plus or minus listeria. It's fantastic work. It's molecular biology, microbiology, and chemistry. And so this is what I spent a good share of today and yesterday doing, and a couple weeks ago, is part of my job is to help interpret those results and to identify where in the processing plant listeria might be found, and then to help with the chemical application to kill it. Okay.
That's
And
remarkable.
I mean, it's pretty fun. I mean, we're on a search and destroy mission, and this system that this company has developed is so slick, and but what's really, really fun is that it goes back to the basic science of ribosomal RNA. And I get goosebumps just thinking about Carl Woese, because what their discovery of the archaea and how the conservation of ribosomal RNA sequences is just it's just so exciting. But anyway, but from a regulatory standpoint, and I'll just tell you, I mean, Eric's very familiar with his story because he lived it too. We had a plant in Rhode Island, and Eric, I got to tell you about that. We had a plant in Rhode Island. It was a 3-year-old processing plant where the FDA comes through and does what's called a swabathon. And what they do is they come through and they take swabs, sterile. They take a sterile swab and they swab a surface, and then they take it back to their lab and they analyze it for listeria. And what happened is in April of 2017, we had 4 hits, positive Listeria monocytogenes in our environment. And it was all around egg and tuna salad sandwiches, which we had manufactured, and a mixer that we had used to manufacture that.
It's 6:30.
Is that okay?
It's Ellie's computer telling her what time it is, so
Okay.
she's making sure she takes her study breaks. So
Okay.
Yeah, my bad. It was
All right, that's okay. So, anyway, what we had is we had these positives, these 4 positives, and FDA does not look fondly upon you if you have positive Listeria monocytogenes in your food contact areas. And so, we did our due diligence and we cleaned it up and we did everything we possibly could to clean it and sanitize it. And then about September of that same year, they came back and retested. And instead of finding 4 positives, they found 12 positives. And that scared the hell out of us as a company. And what we did is we effectively shut the plant down for 3 weeks and cleaned and sanitized. I mean, we were not having very much luck finding the organism, but what the FDA had done is they had actually gone through and they had done a whole genome sequence of the isolates that they had found in our plant, and they were all the same. So the ones in April, the strain of Listeria was exactly the same strain that was found in September. So what they told us is that, okay, you guys have a resident strain of this pathogen in your plant, and it's getting in food. And what they told us then is you have a choice. You call a voluntary recall of the product, or we will mandate a recall of the product. So we called a voluntary recall, and they found Listeria monocytogenes in that product. And then we shut the plant down for 3 weeks and looked for it. Couldn't find it, cleaned and sanitized it, reopened the plant, shut the plant down 4 months later, 5 months later, and sold the plant. And then I found out last week, Eric, Eric was intimately involved because he was leading our sanitation charge on this. He was the one who was helping us identify which chemicals were best to use to help kill this microorganism and to eliminate it from our environment.
Many sleepless nights, not because I was losing sleep, but because I was up at watching the sanitation.
Yeah. So yeah, and yeah, I mean all of us. So what was really entertaining, Eric, is last week Sam Miyota of IEH Labs, he's the president of IEH Labs, called and then sent over the FDA warning letter to a company called Allmark Eggs, and he sent over the whole genome sequence from the organisms that FDA isolated from Allmark Eggs, and it was identical to our strains. So what we were, our environment was probably clean. What was happening is we were getting constant contamination from another source that was coming in. And there's the way Listeria monocytogenes works in an environment is it may be very sporadic. It may from a biological aspect, it'll find a niche and live in that niche. It doesn't compete very well with other microorganisms, but it'll survive well in that niche. And then what will happen is if that niche happens to be in a conveyor, a roller, or a piece of equipment, that a bearing in a piece of equipment, it'll leak out and contaminate the food or the surface that it's on. And it takes a lot of effort to find it sometimes. But that's what's neat about this kit that uses the ribosomal RNA, Eric, is that if we would have had access to that, we would have been able to do a lot more testing a lot faster. As it is, I think we did somewhere 4,000 swabs that costs the company around, about the entire thing, the entire recall and subsequent actions was a total bill of $5.1 million. Okay. And so the number of swabs we did helped contribute to that huge number. But the Listeria right now, the kit that we're using now, would have helped us identify that much faster. So from a regulatory standpoint, understanding the regulations and understanding how to get through with the FDA and how to work with the FDA to get over this shutdown was phenomenally important. And we used both myself and the VP at the company at the time, his expertise, and then we also went outside to an outside legal firm who has experience working with regulatory. And we eventually got through it, but eventually, because of the damage done, because of the reputation of the plant, we shut it down.
So if you had had the ribosomal RNA sequencing, you would have been able to get your swabs back within an hour. Am I
Yep.
following you right?
Yep. We would have, instead of having a 2 to 3 day lag time, we would have had almost immediate results. And, within an hour. And that, what that allows you to do is 98% of your swabs turn up negative. And then that 2% helps you focus in on that location. And you can continue just getting more and more granular in that area and just getting right down to exactly where the organism's coming from. And I think once we got the plant cleaned up through Eric, what we were finding sporadic hits, but they're transient organisms. They might come in on the bottom of a pallet, or they might come in, they weren't really there all the time. So, okay.
You're able to check relative concentrations as well, or is it just for the presence, positive or negative?
It's just positive or negative. The Europeans have a little bit more liberal policy. We have a zero tolerance, effectively. The Europeans have 100 CFU level, so it's a little bit more liberal. How do you kill Listeria? So the way we kill Listeria is we use a baseball bat. Effectively, the idea is that when we in a food processing plant, there are effectively several steps into cleaning and sanitizing the surfaces, the equipment. What we do is we use a caustic compound and clean. And by cleaning, what we're looking at here is we're actually physically scrubbing the equipment to remove any biofilm that might have formed. Come back through with a rinse, and then we come through with a couple different kinds of sanitizers. Now, sanitizers are in many ways just disinfectants. A disinfectant will take almost all the microorganisms off the surface and kill everything. It's almost sterility. Sanitizers knock it down 4 to 5 log cycles. So it's pretty sterile. But sanitizers, you're all familiar with bleach, right? Chlorine, hypochlorite. You're all familiar, you may not be more familiar with things like a quaternary ammonium compound, which is a chemical that is very effective against listeria and molds. And then there's the other ones are such things as peroxyacetic acid, which is hydrogen peroxide mixed with acetic acid or vinegar, and that's very effective. And then there's a couple others, but the ones that we use in food processing are primarily quaternary ammonium compounds, and then also this peroxyacetic acid, which is extremely effective, low pH, and helps while the quat tends to be more neutral. So that's how we kill it.
Do you find that your common strains have issues with mutating?
We don't really do mutations. I think what you're looking towards is something like resistance to antimicrobials. And chemical antimicrobials are a little bit different than biological antimicrobials, like because they're much smaller molecules, and organisms just don't tend to develop resistance because they impact a different system. They may disrupt a cell membrane, for example, which is a common effect. While an antibiotic actually is much more relevant to the biochemistry of the cell. It'll interact with a pathway, for example, and it'll stop the cell growth. Okay, so that's how it is.
Yeah, I think Ellie too. One of the other things to keep in mind with sanitizers is that by changing sanitizers, any sort of resistance that may grow. And Sam, correct me if I'm wrong, but you can by switching and fluctuating your sanitizers, it can actually have the same sort of effect.
Right, but yeah, what the research has shown recently is that there's less the ability. We use fairly high concentrations of these sanitizers. What they're able to show is that at low levels, almost very low parts per million, some resistance may develop, but it seems to be very strain specific, and it's not necessarily. If you hit it with 1 part per million, there may be resistance, but you hit it with where we're at, 200 to 400 parts per million, for example, with quat, it's irrelevant. Okay, that resistance is irrelevant. So I'm looking at a couple of questions here. Undergrad students who will increase their orientation in a research field, volunteer or go talk. If you're interested in something, become fairly well versed in it and understand the actual principles, and then go talk to a professor to see if you can't, volunteer. Even washing dishes. I washed dishes in a fish lab for a year under a work study grant, and even that introduces you to doing work. I have employed a lot of undergrads in my life, and what you want to do is you get your basic hands-on and can prove yourself that you can do aseptic techniques, or you can actually inject a sample into a GC or an HPLC well. And then, what'll happen is, this is how I pursued it, is I always then gave that undergrad their own project, okay? Remember Paul Donlevy? Yeah, Paul worked for us up in Minnesota. Um, Paul actually started out as an undergrad in my lab. He back he actually worked on a project on muffins. When you make muffins with sugar, the water activity is fairly fairly low, so they don't mold very easy. But what happens is if you use artificial sweeteners, because the amount of water that's tied up is a lot less, they mold pretty easily. And what we were looking at is ways to help identify how to increase the water activity or decrease the water activity. So anyway,
So did he just bake muffins in your lab? Was that his role?
actually, if you're in a food science and human nutrition department, what you have is you often have a test kitchen, and so you go make them in there, and then you take them back to the lab and you artificially inoculate them. So you do you do the formulation. That was the time long enough ago that was Splenda was first coming out. Remember the yellow packets that you guys eat? Splenda has a lot of stuff in it that change because it's so concentrated. It doesn't change it doesn't bind up water like sugar does. And so what happens is you get it's a moisture muffin effectively, and it molds much more rapidly. But the other thing is that my wife ran a soybean pathogen lab at Iowa State, and they would get undergrads also, and she'd put them to work. I mean, this is agricultural research but still very sophisticated work, where they're looking at different cultivars of soybeans that would support the growth of pathogens. But a lot of that is just manual labor, where you're out harvesting, you're out washing, you're out doing stuff. But all of that is valuable because that's how research is done. And it's just not you just don't walk into a lab and knock over a bunch of test tubes. There is a lot of stuff. You got to have a reason for doing it. And in biology and biochemistry, there's all sorts of medical applications, and I'll put the bug in that agricultural applications are going to become much more important to us. Everything from drought resistance, which is in plants, to looking at different types of soil composition, to looking at how global climate change is going to impact soil microflora, for example. We looked at how soil compaction during harvesting of trees impacted the soil ecology, the microbial soil ecology, and that took going out into the field, pulling the samples, taking them back to the lab, extracting the soil sample, and then running it through a gas chromatograph to look at the differences. So it is science, and it's microbial ecology, and it's very pertinent, because how we treat the soil is phenomenally important. So, make sense? There's another question. No? Yeah, right. So, orientation, and then current research, Sam. What current research for me is a little bit different, because what we're looking at is we're in an industry, right? And we don't do a whole lot of research. What we may do is we may do challenge studies to see if one floor treatment reduces our risk of listeria presence in an environment. We may look at some of our methods to detect soil on surfaces include using what are called ATP swabs, where we're actually looking at adenosine triphosphate in the soil surface on the clean equipment. So we may look at different techniques to identify that. So it's applied research that doesn't necessarily get published, and but we use it for cost savings, for example. We'll use it for sensitivity. It improves our sensitivity. But we don't do a whole lot of publishable research.
Do you prefer being involved in the research you do now, or versus the research you did when you were in academia?
That's about the same. I never really got the, publishing is one thing, but frankly, you can do a lot of work, really good work, without ever publishing. And I never got the bug for publishing. I look back at some old notebooks and I realized I should have probably published that. I really didn't care. It answered my question, or answered the question we were looking for. And someone else can publish that data. I guess I don't care. That's what it amounts to. So it got the job done. So okay. Any other questions? Yeah.
I have a few. Yeah, if you have time.
I do.
Okay. So if now you're talking to students who are graduating with a degree in biochemistry or biological sciences, perhaps an emphasis in microbiology, something along the way, and they're interested in getting into food safety as a science, but they don't necessarily have specifically food safety as their degree. In your industry, what matters more? Is it important to get a master's, to get a PhD, to go right in and have experience as you're moving up? How do you see the people from the science background in any capacity kind of get into food safety and regulation?
One of the things I think, I do have the PhD, and I used it quite frequently as a beating tool to my lessers, but that's a bunch of bullshit. What I do use I use my credentials in industry right now primarily for regulatory affairs. Saying Dr. Beattie to a regulatory person gives me a little bit more credibility. Okay. But the issue associated with whether or not I'm smarter than someone with a master's or a undergrad, I don't think so. I think smartness in our field comes through experience and life experiences and being able to think. And I guess that may be the use of a master's degree, is it gives you that ability. Some people have it naturally, but it takes a little bit of development to look at a question and work your way through that question and come out with a couple different avenues forward that will solve the issue or solve the question. There's oftentimes people will immediately go to the easiest, obvious answer, which may not be the right one. And so being able to think through that process in a more disciplined fashion, I think that some of the higher education helps with that. So, but some of the people I know in this industry, in the food safety realm, they don't have advanced degrees. They may not even have an undergrad degree, but they are some of the smarter people I know. And but they've got years of experience and seen a lot of stuff. So, that's the thing. I mean, you guys, we're all. I used to teach non-majors biology. Okay, 450 students a semester. 12 to 20 TAs, and with all the lab sections. And what every year before I started the class, the thing about non-majors biology is people walk into the class and they're terrified of it. Undergrads are terrified of that class, and you take the TAs down to the creek, and you make them squish through the mud, and you make them look at the stuff in this mud, and you make them look at stuff. And this is what you did when you were 7, 8, 9 years old. It was so cool back then. Let's bring that gee whiz back. Let's bring that observational characteristic back to these students who are afraid of this course. We don't want to make them hardcore biologists. We want to make them understand the principles of observation and how cool this world is that we live in. So, some of the brighter people I know can take and look at a situation and make conclusions about it or make observations about it that help lead to a conclusion. And that's where that's where the advanced degrees may come in to help. Okay. Okay. So anyway, what else? My son just popped his head in. Sorry.
I personally have a question about your experience getting your PhD. If your first research topic, I guess, that you were working on with the oysters, if that hadn't lost to funding, would your PhD have been under a different subject?
Yes, quite a bit, and probably my trajectory would be different. Okay. Um, I probably would become more of a biochemist at that point. Probably more of a molecular biologist. We were doing things back then that were pretty cutting edge. I mean, isolating messenger RNA, even now, is extremely difficult. We were developing methods on ways to get message out. There's a lot more message in a cell than there are enzymes, and so getting that information out and how to do it would have put me in a different trajectory. Okay. That was 1985, 86. So, that's a long time ago. I'd probably be in academia and poor. So, that's how it works. So.
What did that mean for you as a PhD student to lose your funding? Did you have to go find a new advisor and start the process over?
I did, but I tell people that I did my doctorate in Squirrel's Tavern in Corvallis, and because what we did back then, it was acceptable to go out and drink with your professors, and we did a lot of that, and so everyone knew the struggles that I was having, and knew that the funding was probably going to dry up, and this included a couple of very important faculty in my life. So moving over to a different lab was okay, and they knew, but it was the writing of my own grant, or writing the proposal that got me the grant. I think that helped a lot. So I effectively could have worked in anyone's lab at that point. Okay.
So the connections you made while you were there had a big impact on.
Yeah, they did. Yeah, and even I mean, yeah. For a lot of my professors are dead now, but that's the way it goes. So. And what else we got here? If a student lives in a city with a well-known company for research, how can she take advantage of that? That's a good question. What you could do if you do have a company in your city, figure out how to apply for a job. I guess that's all you can do. Most private companies won't take volunteers very readily. It's an insurance issue. Okay, so you get hurt on the job, they're not sure if they're responsible for you or not. But if you are, in fact, if you have some skill sets, even if you go in as a dishwasher, it's not a bad thing, because it does give you that experience, and that's showing initiative on your part. It may lead to other things within that company. So I would encourage trying to get in, even if it's the lowest level. You all heard about like the mailroom clerk who becomes the CEO of the company, right? But I don't think that happens very often, but it certainly will get your foot in the door, and then perhaps you can go on from there. Oh, no. As you get more experience. Plus, if you've got, I know you guys are all distance people, but if you've got a professor who's helping you with any of your courses, you might put out a query to them and say, do you know anyone in my area that might be willing to take on, a student, or can you, use those connections to see if people won't, help you. That's what we all like, helping students. I mean, that's why we were in this field. Well, some of us at least, but you're our progeny, so we want you to be successful, and so you go off and do good work, for private industry. It looks good on us too, or it looks good on us, I guess.
I believe also, just to toss in another note there, that there are internships available. Career Services does actually do some with internships. They hear internship. Another possible avenue to even just get into some various aspects of industry in a lab.
Sure. What did I look for when I was selecting undergrads? People who are curious. I think that's really critical. It's research is curiosity, right? And so it's you had to be pretty motivated. My last grad student, she and her husband were from Russia, and Dilly would Diliara Isanova. She hard worker, but she wore stilettos almost every single day, and it was really entertaining because she walked from married student housing almost a mile to the lab in Iowa winters with snow blowing and in stilettos. It was just, she had the dedication and the drive to get to work and to do the work. And that really valuable to me. So, how do you recommend conveying? I'd just come out and just flat out say it. How do you put curiosity onto a resume? I think you use words in your description of yourself as inquisitive, curious, wanting to know how and why type things. You can, and if you can develop some examples of where your curiosity led you someplace where you didn't expect it to go. Okay, or how you got stuck in the box because you were inquisitive about what was in the box type thing. Those are the kind of things that I think companies want to hear. If we were an accountant, they'd probably not want that, but we're researchers, guys, or we're in sciences, and that's what science is all about. It's that curiosity, and showing the prospective employers or people that I do have a certain drive to discover. Okay.
And if you do see a cat in the box with you, get out because it's Schrodinger's.
Yeah, right. No, I think yeah, you guys are all in this because you're interested in something, and use that curiosity as a point in a resume. I think inquisitive is the word I would use.
Thank you.
Yeah. What else we got?
I mean, if you're good for it, there's a couple more as far as the industry specifically. Do you find that there's a cap as far as how far you can move up within the industry without more education, or can you go further just on your experience alone?
You can go as far as you want with your experience. Our new CE or COO he's about my age, and he's got decades of experience coming out of something that's entirely non-related to the food industry. So you don't have to necessarily, you have to be curious. That's the curiosity end of it. You have to be looking, you have to be able to show that you can perform, and that you can perform well, and be accountable for those actions that you take, with full justification for why you took the action. In our fields, the food safety and quality sciences, what you have to do is, I made this decision based upon this journal article, this guidance document, and I'll stand by that decision again. And people appreciate that honesty and that accountability. Okay. So, and that is what will drive you in the industry. That will drive promotions. Okay.
It's a valuable perspective because we're at the undergraduate, college level where they drill education. You want to be successful at all, you got to get all the letters after your name. It's really
Yeah.
valuable to have the real experience of somebody who's like, not necessarily.
Yeah. I don't think it's necessary. And you guys, I feel bad for you guys. And I felt when I was a hardcore teacher at the Chico State in California. The number of undergrad general education requirements that you guys are required to take is stunning to me. When I was an undergrad, we had 9 units of general ed requirements, and now you guys, half your life in undergrad is spent taking useless classes, in my mind. You're not allowed to. I took physical chemistry. I took, you know, engineering courses because I was interested in them, and they did help in the long run. I did poorly in them, but they nonetheless helped in the long run. So.
I think maybe one other side note I'll toss out there too is, when it comes to getting the alphabet soup, so to speak, after your name, there are plenty of professional organizations that do offer certifications which are validated and true. And ASQ, for example, American Society of Quality, RAP, which is Regulatory Association Professionals, or I believe something along those lines. So there are other places, and Sam, you might have some others that you can think of that are like International Food Technologists, for example, where you can get additional certifications, and through that you have those, like I said, the alphabet soup at the end of your name.
Indeed, and it's yeah. I mean, uh, some of them are useful, and some of them are nothing more than salad. So yeah. So all right. What else? Anything else?
I think that's everything I had on our pre-list we came up with. How about you,
Okay.
Eric? You talked about that Sam might be interested in talking about new assays and identifying particular microorganisms.
Well, I think with the new assays, what we were talking about earlier with the ribosomal
Yep.
identification of listeria, that would be a prime example of new tech that has significantly impacted the industry, as well as improved our ability to go investigate issues when we find them.
And it made our food safer. Okay, so it's I think that as more of these come out, we will, it'll be we'll find it safer. What I had a discussion. I had a discussion with Andy Hearns, who was our past VP. One of the, being able to understand how tests work is really important. So the USDA, the FDA recognizes that there may be levels of allergenic proteins in foods that do not cause a reaction. Okay, USDA does not recognize that. They have a zero tolerance. And what happened a couple of years ago was Newlywed Foods produced breadcrumbs. Big deal. But they also produced a breadcrumb with dairy or milk in it prior to the other breadcrumbs. So there may have been residual milk allergen in this other set of breadcrumbs. Okay. So, and we got caught up in that. That eventually became a recall. But what happened is FDA recognized that the amount of milk allergen was so low in those other non-allergenic breadcrumbs that they really couldn't call a recall very well. Whereas on the other hand, USDA made Tyson's and the big people who make chicken nuggets and things that are coated with flour or with the breadcrumbs, they made them recall millions of pounds of these items, okay, where there was no food safety risk. Fast forward to this week, we've been having discussions on what tolerance level are our allergen assays are our allergen assays on our that we use to detect allergens on equipment, are those sensitive enough for USDA purposes who have a zero tolerance? So our test kits currently go down to about 3 to 5 parts per million. And that's, I mean, that's milligram quantities on a 12-inch square surface, and phenomenally small. But that surface, if it registers no detectable, uh, no detectable, is is are we sure that 3 parts per million, below 3 parts per million, if 2 parts per million exists and it's on the surface, is the product going to be free from that allergen? And that's the stand that USDA is kind of taking right now. That, well, you guys don't know below your level of detection if you're free from allergen or not. And so we're all kind of scrambling on that. And, we want to be certain that we're in good graces with regulatory, but also that we're not doing something futile, and that we're cleaning to the level, we're microbiologically safe, and frankly, we're protein safe, also allergen safe. So I don't know, it's that's an example of new technology that has helped us, but also could put us into a bind.
Yeah.
So,
I would imagine, particularly if the technology advances ahead of the regulation,
yep,
if you develop the technology to detect a more sensitive parts per million,
yep,
then does the regulation have to change? Are you now in contamination if you meet that lower level?
right. And frankly, that exists already.
Yeah.
We can test down to microgram quantities we can go down from milligram quantities down to microgram quantities. But the relevance of that is very low. Okay. So, the test kit companies recognize that at the lower levels, we don't need to go much lower than where we're at now. Okay. And this is coming from a father who has a 15-year-old who's acutely allergic to peanuts. So I'm erring on the side of um, reality rather than caution. So, okay.
What out of my own curiosity, the people, because it sounds like most of the actual development in the industry is coming from the private sector, just based out of need as you're developing and improving your own standards and your own I don't know.
Yeah. No, you're right.
What positions of people are actually actively working on this, on developing these solutions, these new testing assays and such?
Oh, certainly what we see is emerging pathogens. Okay, so what we recognize is that, for example, what I expect in the next couple of years is a rapid method for norovirus. You're familiar with cruise ship virus, right?
Mhm.
It's the leading cause of acute gastroenteritis in this country, and most of it comes from eating someone else's poop. Okay, because what they did is they failed to wash their hands when they had diarrhea. The story I like saying is that when you're in the acute throes of diarrhea with 24-hour stomach flu, what you're doing is you're producing every gram of your diarrhea has around somewhere between 2 and 8 billion norovirus particles. The infectious dose on norovirus is 17 virus particles. So a quarter cup of your diarrhea when you have acute gastroenteritis could make every person sick, every person in the world sick with your virus. Okay. So what I expect we'll see over the next couple of years is we've identified the issue, which is norovirus, a significant problem. What we will see is we'll see a rapid method for detection of norovirus on surfaces. Okay. And so what happens is the epidemiology drives some of the the research to solve a problem. You familiar with cantaloupe? The recall on cantaloupe, there's been several over the past couple years. How do you wash or sanitize the surface of a cantaloupe, which is rough surface? How do you, and you go back to lettuce or spinach, how do you sanitize, how do you eliminate all the microorganisms that may be present on that leaf? How do you do that? Because, you know, from an agricultural perspective, we can't prevent everything that flies over the field, slithers through the field, lands on a leaf and defecates. We can't prevent that. So, and then we also know that you know, the microflora of a leaf, you know, those organisms are phenomenally difficult to remove from that leaf. So, you know, how there'll be new interventions that'll come from the agricultural side to eliminate the microflora on those agricultural products that we eat raw. Okay, so it's you know, I hate to say it, but it's the illnesses driving the research.
You're feeling need.
Yeah, you feel a need. That's exactly, solve the problem.
It's remarkable. Thank you for sharing.
Yeah, not a problem. Okay. All right. Well, I'm kind of talked out, so I got family banging on the door. So, are we good?
We're great. Thank you, Dr. Beattie, for coming and speaking with us and sharing.
No problem. Yeah, it was pleasant. It was a pleasure, guys. Thank you.
Thanks, Sam.
Sure. Eric, if there's any other questions that they ask afterwards, let me know.
I'll get them over to you. Absolutely.
All right, thanks. All right. Bye-bye.
Bye.